Good morning, everybody. It's a great honor and pleasure to be able to be chair of this session. It's also somehow a little bit personal for me as well. We're going to hear the talk by Haruo Naito, who's heading Eisai since 1988. He started in 1975, and then he took over 1988. He was in several positions till then. And what he did is actually something which is really amazing. He made out of this company, which was a very successful, mostly Japanese-based company, a truly international R&D company, which is present on a worldwide level and is expanding massively throughout the world. And one of the reasons of the success, I guess at least, I mean, this is my psychiatric point of view, is that the company came up with Aricept, which is really changed the game in the Alzheimer's field, and it's still, as far as I know, the most sold Alzheimer's drug worldwide since 25 years or so. So why I think it really changed the field is because, just on a personal note from my side, as I started in psychiatry mid-90s, there was no treatment and there was no possibility to do a positive diagnosis of or probable or good, somehow good diagnosis of Alzheimer's disease. And it was totally neglected. I started, actually the first patient I saw myself was actually Alzheimer's patient, or patients doing a clinical trial on a substance at the time which obviously failed. And it was a totally neglected field. Nobody wanted to deal with these patients. And then these treatment options came out paired together with diagnostic possibilities which were better and better and better, ending up in, nowadays being just before the launch of peripheral biomarkers, which will come up in the next 1, 2, 3 years on a larger level. So it's one thing. The other thing is that the company was still working in Alzheimer's disease and came up with a first really working antibody, lecanemab, as a disease-modifying antibody for the treatment of Alzheimer's disease, which is approved in US and it's just about to be approved, for example, in Europe, also in China approved. And it's going to come on a larger world level, or it's just beginning to come on a larger world level. But it's, it would be wrong to reduce, the merits of the company only to Alzheimer's disease, even though for me, it's like the key point. But the company has drugs in many other areas, especially also oncology, but also is committed to rare tropical diseases, neglected diseases, and many other things like drugs for insomnia and so on and so forth. So it has an increasingly large portfolio, but of course we are probably here most interested in what you're doing in Alzheimer's disease, but there are also drugs for epilepsy, drugs for ALS which are developed, and so on and so forth. So warm welcome from my side. Thank you for coming.
Yeah, thank you, Dan, for just an excellent introduction. And he covers most of my part already, so I may finish now. But this is a great chance for me to speak in front of you this time. This distinguished scientific conferences, which has a long history of a contribution to psychiatric treatments and options creation to the patients. So it's my great honor today, as Dan talked about, we recently had some success of developing a new Alzheimer's drug called lecanemab, which is removing A-beta proteins from the brain and showed quite clinical meaningful results. So through those processes and steps, we have a lot of experiences, which is just something positive or negative. But anyway, those experiences are kind of invaluable learnings for us. So I'd like to share some of that. And later part of my presentation, you may be interested in how the price of this kind of new innovation would be set forth. Might be not your daily interest, but we also did somewhat unique job for pricing of this drug. So I'd like to talk about that in a later part of my presentation. So let me start. Okay, I have no COI. So lecanemab discovery and development. Next, please. So the origin is back 92. You may know Professor John Hardy at University College London start advocating the amyloid cascade hypothesis. And he said this is the one of the primary cause of Alzheimer's disease, and that is this. So from A-beta precursor protein, enzymes such as gamma secretase or beta secretase would carve out A-beta monomer, and those monomer become oligomer, soluble oligomer, and the end of this cascade it's going to be end up by insoluble amyloid plaque and deposited in the brain or the brain vessels. So this cascade is the cause of Alzheimer's disease, so-called A-beta hypothesis. So this hypothesis, stimulate a lot of, the scientists or pharmaceutical industries. Oh, it's good. We may create a new drug based on this hypothesis. So many attempts are repeated, like approaching those monomer production between here or the very end of insoluble plaque removal. So many attempts has been made. And we are also very interested in this cascade goes two ways or not. Still, there are several arguments to clarify furthermore what this cascade means. There are still some of unknown part. We work on this monomer part by secretase inhibition, such as beta secretase or gamma secretase modulation. That attempts has been failed. Cascade didn't move to this direction. Even we reduce monomer, those oligomers or plaque is not moved, significantly. So this is the very starting back in '92, and 2001. You may have heard of Professor Lars Lannfelt of that time Uppsala University in Sweden had studied the families live in northern Sweden, Alzheimer's family, for many years, visiting those villages, regularly and understanding their life, try to collect the samples from those families. Those long-term efforts by Lars, finally he found on those families there are one mutation at the 22nd amino acid mutation. And since those families living in the very cold place near North Pole, he named it the Arctic mutation. And through the study of deaf brain of those families, he also found a large amount of protofibrils. This part, A-beta protofibrils, has been produced in the brain of those families. So Lars made pretty substantial preciseness over this A-beta hypothesis. Now he starts saying, actually, neurotoxin for Alzheimer's disease is not maybe those plaque fibrils, but protofibrils. And this can prove the disease is progressive, because we know even if we remove those parts, the disease never can stop. It's going to be proceeding. So this protofibrils as a major neurotoxic agent kind of hypothesis has great significance. And we've been very much attracted by his hypothesis and start working with them. And now we work to find out specific antibodies which could be targeting protofibrils and remove the protofibrils from the brain. And we finally find out antibody specific to A-beta protofibril and named it lecanemab. That was back 2007. And recently, 2 years ago, we finalized the final study, phase 3, very successfully. I'll touch on later. And the drug is available now in the United States, Japan, and soon in China. And we are now filing the regulatory approval for 26 countries right now. Next. So this is a snapshot back 22, John Hardy, myself at the University College London, because we've been collaborating with this group over 10 years in neurosciences, and John Hardy is part of that collaboration. Next. And Lars Lanfeld, this is a snapshot at 23 Boston CTAD meeting. CTAD is Clinical Trial Alzheimer's Disease, one of the most top scientific meeting for AD, and we celebrate the success of the phase 3 study at that time. Next. So one of the lessons, number one, is academia collaboration. Of course, without John Hardy, without Lars Lanfeld, lecanemab will never coming out. This is a fact, but also very strong rationale supporting academia collaboration is essential for drug discovery now, because almost all drug discoveries are now based on human biology. Used to be we study animals, or brilliant scientists, serendipity may be the major cause of the innovation, but it's all gone. Absolutely and definitely, the things should be all based on human biology itself. So genomes, genetics, or whatever the information, biomarkers coming from human samples. That is the base of drug discovery and development. And then where are those high quality human samples located? It's not in our industry's hand. but the most probably academia or research institutes has a chance to have such kind of a high quality human samples. Therefore, that is a major reason drug discovery should be collaborating with academia. And how quickly we can formulate hypothesis based on human biology and validate that also based by human biological method using IPS or many organoids techniques these days. So first lesson we learned from lecanemab development is academia collaboration is the key. Next, please. So phase 2 study, we call it study 201. Next, please. So this is a large study. Actually, almost 900 patients entered. Target population was early Alzheimer's disease, namely MCI due to AD or mild AD. The major purpose of this 2-1 study is dose finding. As we all are aware of, that antibody targeting brain, most difficult part of the drug development is dose finding. We need to find out the exact optimal dosing, which could be, leads to the brain target, should pass through BBB and leads to the target. So dose finding is the most sophisticated and complicated part of the drug development for brain targeting antibody. And we actually compare those 5 dosing with the placebo. And also the matter is the endpoints, clinical endpoints. How are you going to evaluate the efficaciousness of the drug? So endpoint is another key area. And for this 2-1 study, the primary endpoint was outcomes at 12 months. Outcomes is a composite score which Eisai developed good to identify cognitive functions in early AD patients. So we use that, but we like to examine it at 12 months. Relatively short period for AD treatments. But simultaneously, we have several secondary endpoints. All of them are at 18 months. So the study can continue over 12 months to 18 months. But we set forth the primary endpoint is at 12 months. So what's happened next? Actually, this study took 6 years to finish. Extraordinarily long time. Usually, this kind of phase 2 study, although it's complicated, but we'll finish by at most 2 years maybe. But it took 6 years because it's completely new study, and dose finding has been so difficult. We apply most modern Bayesian statistical technologies to allocate the most next patients to most promising dosing option, but still took 6 years, very long time study, and the study was failed because this primary endpoints, outcomes, 12 months didn't meet. Namely, we cannot find out any statistical differences at this endpoint at 12 months. But thank you for the secondary endpoint. We set forth the second endpoint at 18 months. So if the treatment duration is to 18 months, we hit 18 months outcomes. We find out statistical significance. So although study has failed, but we found if the treatment continued to 18 months, it might be successful. So I believe that this 2-1 study, the key studies in the development of lecanemab, because although it took quite a long time and failed study, but we acquired those critical information for the next phase 3 study. One is the hypothesis, namely A-beta protofill hypothesis, could be the right hypothesis because we see the significance differences over the placebo at 18 months. And the target population of early AD probably is the right population. And particularly important is that we found the right dosage among those comparison. The 10 milligram per kilogram biweekly, no titration is the optimal dosing schedule for this drug. And the endpoint should not be at the 12 months. And we decided to pick up a more universally, used endpoints, which is a CDR-SB as the final endpoint. So 2-1 study take long time, failed, but we acquired those 4 rights, most crucial information for the next stage. So our lesson is, should not proceed until we can confirm the rightness of essential part of the business. Haste makes waste is also true in clinical development. This is a lesson 2. Next, please. So phase 3 study called the Clarity AD study. Next. Study design is become very simple now. We are again targeting early AD patients, but almost 1,800 patients are enrolled. It's the largest study ever made in Alzheimer's disease in the world. And now we know the 10 milligram biweekly is the right dosage, so only one arm, one treatment arm, and placebo arm, 1-to-1. Direct comparison. And the primary endpoint is 18 months CDR-SB, not 12 months, but 18 months CDR-SB was the primary endpoint. By the way, the period that we conduct this study was peak of COVID-19 pandemic years. So we used a decentralized clinical trial with the agreement of the major agencies. So remote diagnosis, home visiting, remote cognitive assessment, and safety monitoring has been conducted, for this study. But study is not delayed; it's almost done on time. So some special efforts made due to this. Next, please. So the results were just remarkable. Primary endpoint CDR-SB, it's a cognitive dementia rating, sum of box. Physicians can check 6 different aspects of a cognitive function of the patients, and adding them all made a score. Has shown that 27% slowing the decline, and compared to placebo, p-value is 0.00005. There are several people who are familiar with this clinical development, but usually, agency approve the drug if p-value is less than 0.05. And if it's less than 0.01, it's called a statistically significant, highly significant, results. So compared to that standard, this is just enormous differences. In other words, what it meant is if we repeat the same study for 200,000 times, 999,000 times we got the same result. So in other words, this shows that this result is true. All secondary endpoints showed similar results with a high statistical significance. We are particularly interested in this part, quality of life endpoints, because those endpoints are measured by caregivers who've been with the patients for almost all times. They evaluate how the a difference would be observed in the daily living activities, and the magnitude of effectiveness has been much larger than cognitive function, and also showed a very high statistical significance. The lowest part is ARIA, is amyloid-related imaging abnormalities, edema or slight hemorrhage, but it's all the range of what we are forecast beforehand, and also almost all majority of those ARIA is asymptomatic, and occurring the early part of the treatments, and very good monitored by MRI. So those ARIA occurrence ratio is also very acceptable, I would say, much lower than the other antibodies. Next. So history maker in modern drug discovery has been made by lecanemab, we believe, because of all endpoints clear, but not just this, but we conducted subclass analysis with the agreement, predetermined subclass analysis with the agreement with the FDA. And what kind of a subclass? One is the concomitant usage of, let's say, Aricept or memantine, those AD symptomatic treatment or not, and there's no differences. All two groups, the drug works. In clinical stage, subgroup MCI, mild dementia, also both groups, drug works. And ApoE4 status, carrier or non-carrier, it works. And geographies, 3 geographies, North America, Europe, Asia, it all works. So the results was very consistent over those subclass analysis. So FDA and CTAD reports, specialists judge the drug is benefit risk positive and clinically meaningful results. Next. This is the picture of a presentation of the results at CTAD back 22. A great moment for all of the audience together. And on the same day, a publication made at New England Journal of Medicine on the same day. So it's dual transparencies of those results. Next, please. So let me talk about slightly more detail of the data. This is a prediction based on clinical pharmacology modeling before starting phase 3 studies. This chart is try to predict what's happening with the amyloid PET scanning would be look like. And this data showed if this dosage, probably around 1.5 years, 65% of patients amyloid status by PET scanning would get to be negative. So this prediction has been made by clinical pharmacology modeling. And also major indicator endpoint, CDR-SB forecast, also biweekly dosing, the around 18 months, average information, probably something slightly less than 30% reduction in CDR-SB decline could be possible. And amazingly, those are the forecasts for ARIA, side effects, because through those studies we know this ARIA is related to Cmax. So our scientists had prediction ApoE for homo, hetero, or negative case ARIA occurrence ratio could be like that. Okay, next. So this is the actual results. And we've been amazed, and you may be also amazed, how close those figures to the prediction by clinical pharmacology modeling. 67% negativity, the modeling suggests something like 65. 27% stop worsening, and the model suggests slightly less than 30. And those ARIA occurrence are very, very close. So, good clinical pharmacology modeling is another lesson we learned, because by knowing that, firstly, we feel very better, because those phase 3 is almost a $1 billion business. Conducting this size of study needs to use up about $1 billion easily. So before that, we are relatively feeling good, probably with success. And also the actual results are very similar. That suggests in the future maybe we can be somewhat eliminate phase 3 study, or even downsize the phase 3 studies. So this particularly emphasize early part of clinical studies are so important now, because of those clinical pharmacology technologies are rapidly progressing. Next. Success elements number 4 is person, scientists. Particularly, I like to talk about the top leader of those projects should be having those kind of qualification. One is who can make a final decision. Otherwise, there are several people who don't like to make decisions. And those people cannot qualify to this. So right or wrong, I hope it's right, but anyway, right timing, that person should make decisions. So this is a requisite of the leader for this kind of big project. And just concentrate on this. Don't think anything others. Only every time, 365 days, concentrate on thinking about lecanemab, thinking about this study, thinking about the patient, thinking about the progress. That is needed. And however, there are always failures, of course, in between. Final success, before that, there are lot of minor or sometimes major failures. Regulatory discussions in between, sometimes deadlocking, stop the trial for a few months easily. But those people even failed, never giving up until to be successful. So they said, Mr. Naito, we keep on failing until to be success. I don't know what it means, but those mentality is needed as the leadership of the project. And having the courageous enough to look back at what they did wrongly and try not to repeat that. So those might be a kind of characteristics we need for the one top leadership of this kind of project. Then, next. Also, we need stars. This is a kind of technical background we need, such as a non-clinical biological pathway, biomarker, clinical pharmacology, statistics, and regulatory. Each of those areas, I can easily identify who is the leader by first name. So the visible professional with specialty is number 5th, kind of elements. Next. There's a picture in the middle center, Dr. Lynn Kramer, who is the exact such kind of person leading all of the projects. Chief clinical officer of us, had a lot of experiences, but the lecanemab will not be succeeded without his leadership. Next. So, failures, number 6 elements. As Dan talked about Aricept, thank you very much for that. And that was back 97. We launched the drug in the United States. And since then, of course, all of our stakeholders are looking at us as what's next. We expect you can succeed the next treatments. So we made the whole effort, gamma secretase modulator, beta secretase inhibitor. You can remember that A-beta cascade for earlier part, all failure. Particularly this one at the middle of phase 3, it's ordered to stop. And most recently, aducanumab. This is a collaboration with our partner Biogen, and Biogen's own development, and we are, partnering that, paying half of the all expenses. But aducanumab has finally not been accepted well in the U.S. community, and we need to terminate that aducanumab project. It was quite a pity. But finally, we reached to this lecanemab success. Okay. So why we need failure? Actually, this is a chart of Akashi Channel near Kobe, the most tough part to navigate the ship, because current here goes to opposite direction morning and evening. And, there are a lot of dangerous spots, rocks or, deep, not deep, those things. So if you're a captain and navigate the ship to the goal safely and quickly, you better have a very precise chart by numbers like this. And Alzheimer's ocean, we only put up the figures by experiences or hopefully by success, but in many cases by failures. So now with those failures, experiences, we know how to navigate our ship to the goal, reach the patients with the successful regulatory status quite well, I believe. Of course, not perfect. So Alzheimer's disease is thought to be kind of red ocean, very difficult and complicated, busy areas. But for us, AD is kind of blue ocean because of we know the chart now. Next. So let's talk about the pricing of this lecanemab. It's a kind of innovative, truly innovative drug, and societal meaning is very great because we know negative impact to the caregivers, mainly family members, are enormous. They lose the opportunities to work, reduce the working time. Those burdens are somewhat saved by this lecanemab treatment. So how are we going to price this drug so that people can understand that is a big issue for us, and we've been preparing for probably past 3-4 years before the launching. So let me talk about this. So firstly, you may amazed modern industry, not just pharmaceutical industry, are now strong premise to tackle on social issues. So you may think we are a kind of profit makers, but profit making is good, but that could be only possible after we feel our social responsibilities, or finding out the solutions for societal programs. So this is probably 80% of the CEOs, particularly in pharmaceutical industries, looking things like that way. So creating impact means solving the societal problems, giving the solutions to the patients, a positive one, we now called simply impact. And so impact creation is a major goals of the industry now. And we like to talk about it more by comparison to the last year, how impact enlarging or unfortunately diminishing, or what Eisai's impact creation is, let's say, compared to Takeda, how large is that? This is just example, no intention, don't misunderstand. But so this kind of purposes, we need to be quantified, this impact in hopefully monetary value. So impact could be health, safety, quality of life, or peace, or whatever, those intangible, usually not shown in how many dollars or how many yen, but to compare to the other companies or try to get a track record of impact creation, better to be, figured. Next. So for that, we need to put up a price tag to Alzheimer's disease. What it means? Simply, how much would you pay if you are free from Alzheimer's disease for 1 year? I don't know, Dan, how much you pay for that. I don't ask that because it's certainly controversial, but there are several studies, good studies, doing this, and those studies are calling willingness to pay threshold determination. So usually the range is GDP per capita of the country 1 times to 3 times. And some studies, classify the disease to the scale, relatively now easy to treat disease to the very tough disease now. So those tough one, probably the highest willingness to pay threshold should be applied. And those relatively now common for treating would be lower threshold application. So this is a logic. Next. So we think this is a great idea, but we need more precise data to that. And what we found is Archimedes condition event simulation model for Alzheimer's disease. It's a disease simulation model. So what we did is, ADNI data is a cohort data of the United States pick up probably about 2,000 cases, which is similar to the Clarity AD trial target, and divided it half, and Leqembi, namely Leqembi treatment case and standard of care case. And for Leqembi case, we extrapolate the clinical study results. And simulation runs for, for each life of those cases. Okay, what's happened? So this is an equation to calculate impact. It's complicated, but rather simple. Let me try to explain that. 0.64 is Cori gained. So key is Cori. And what is Cori? Is length of life times QOL of each year. So if you live 8 years as EAD patients, what would be your quality of life in second year, third year, fourth year? We have some basic data for that. So the end, Leqembi treatment group get 0.64 more Cori gained than standard of care group. And this is, in other words, Cori is the value of health outcomes as a whole. And standard care case, that Cori was 3.7, and the Leqembi case, it's added 0.64 on that. That means overall health outcomes increased almost by 17% in the United States. And interestingly, we did a similar study for Japan, and this Cori increase, although figures are different, are also 17%. So now we know this Cori gained, and willing to pay threshold, we apply the highest one, $200,000 in the United States. So this is a very core of Leqembi impact in money. 7,000 something is the cost offsets, caregiving cost offsets, and the medical cost offsets. Adding that divided by time on treatments, average time on treatments, and we got $37,600 per person per year is the impact we create in the United States. Okay, next. So what we did then to the price, we determined, firstly, this is 10 years time span, and we estimate how many patients could be administered by lecanemab. So total number of patients in 10 years times this impact is the total impact in the United States in 10 years. And we decided to get back 60% of that total impact to the public, because the US has a lot of social infrastructure of healthcare, such as Medicare, Medicaid, and those things, and we like to support that. Public stakeholders are patients, families, caregivers, medical professionals, payers, and governments. But 40%, the rest, please allow us to return that to us as the rewarding the innovation for us. Namely, Eisai shareholders and employees would be like to receive 40% of this Leqembi total value. So we have now our portion of impact in money. And from that, we can reach to the annual Leqembi, the brand name, lecanemab cost in the United States would be $26,500 per patient. So this is a value-based pricing. And we explain it, and those results are published in a few papers simultaneously. And I believe in the US, people, particularly payers, understanding this pricing and this pricing, mechanisms as well. Okay, Next. So I'm reaching to my final part. That is, since RCEP research, we continue AD research almost for 40 years. And how you can continue the same research for 40 years? And answer is very easy. We've been, socializing with the patients during this time and understand the patient families' pleasure, anger, sorrow, and joy, and also we know their expectations to us. So our employees, something like over 10,000, are requesting to spend almost 1% of their business time to be with the patients or to have experiences, the same experience with the patients. And so it's a kind of empathy, intersubjectivity status with the patients and Eisai people. And we know patient families like to see this kind of future. AD treatment option will progress greatly by the drug based on AD true pathologies. And the day AD becomes curable will surely come. So this is the major reason why we are keep on motivated and continue this research. Next, please. So this is some of the pictures of socializing the patients. So you may be amazing to look at, they are either smiling or crying. I mean, our people. So this is about one day, 2 days after start, socializing. Of course, language is not useful. Boundary or anything like that exists in those behind the smiles. This is a pediatric cancer ward. One of our managers in charge of Lenvima oncology drug is just crying to say bye-bye to the boy and a bunch of others. Next. This is a recent socialization case of myself at our place. Those people are having their homemade soaps. And we have a kind of selling shops to sell those soaps to Eisai people, and we are enjoying that, having a very enjoyable time together. Next. And this is a recent shot with early AD patients with his grandson, already start treated by lecanemab almost 3 years. So thank you very much for listening. That's all my presentation. Thank you.
Thank you so much for this absolutely amazing and fantastic lecture. The talk is open for discussions.
By the way, if you need a slide, it's going to be available, so please ask a secretariat.
So, any question?
Hi, Jordan Karp from Tucson, Arizona, United States. Thank you for a beautiful talk, really transparent and informative. How long will Leqembi be prescribed for patients? Is there an end date, or is it 3 years every other week or for the rest of their lives?
Yeah, thank you. That's a very good question. Now we are filing the dossier for maintenance dosing, which is a half dosing. So maintenance dosing means we need the initial period. So how long should be initial period? 1 year, 18 months? That we don't know. It's going to be subject to discuss with the agency. So we still don't know. But at least at this moment, we have a very solid clinical data covering for almost 36 months and showing the efficacious results during these 36 months. And as this is a removal of one of the pathologies of AD, so as long as you are treated, the effective size is looking like enlarging as the AD-DMT original characteristic. And also, it's apparent AD is a progressive neurodegenerative diseases, so we are saying the continuous treatment is good for the patients. And we are now developing a subcutaneous auto-injector, which could be injectable at their places. No need to go to infusion center anymore. So those are the current status.
Good.
Am I answering
Yes,
you?
perfect. Thank you.
Hi. Hi, I'm Brad Miller. I'm from Columbia University in the U.S. Excellent talk. It seems like the antibody does an amazing job of removing the amyloid from the brain, and it had a highly significant effect on symptoms, but the symptoms seem to be delayed. They weren't stopped. And so if you can remove amyloid so well from the brain, but the symptoms continue, what do you think the next necessary targets or approaches are going to have to be to stop the symptoms from progressing?
Yeah, thank you. Good question. You may remember phase 2 study, 201 study has open label extension studies after finishing that. And there are some 2 years gap of no treatments period in between. So during that period, 2 years, people get worse. A-beta deposition gradually has started. We confirmed that by biomarker, blood biomarker behavior. So yes, we need to don't stop the treatments. So continuous treatments, even lower dosing, is one necessity. Second is the starting the treatment when. So RCEP day, it was early AD. And lecanemab, it became MCI, so one stage earlier. And we are doing a preclinical study, which is the, almost no symptoms, A-beta accumulation started in a very early stage. We still had a study. If that's successful, we can, even one stage earlier start treatments. So early starting, the more efficaciousness you can expect. So continue to treat and try to get the starting early as possible by furthermore clinical investigations, trials. Those are, for us, suitable solution at this moment.
I think this is a very, very important point. At the time point when peripheral biomarkers are going to be generally available also to GPs, we're going to face a lot of preclinical Alzheimer's disease. And in my opinion, this is also the most promising population because they don't have scars in the brain, or not so many. So I'm glad to hear that and I knew that you're doing also preclinical Alzheimer's disease, which makes total sense. When are we going to get results? The study is a long-time study, and target number is 1,400 patients enrollments, and we originally think it's maybe very tough to recruit the patients, but it's going very well. Patient entry is much, much earlier, faster than what I'm expecting. So, sorry, I don't tell the exact timing, but it's going very smoothly. And as Dan, you talk about, we use blood biomarker very substantially in that study. But would it be like, 2 years, 5 years, 10 years? But, somehow...
Yeah, it's
roughly.
in a few years. In a few years, we will get the result.
Because if so, we're going to face these patients before your results. So what would we do with them? 45 years old, otherwise healthy, clear Alzheimer's disease, but no whatsoever clinical manifestation. So that's going to be, I guess, more and more a population which is hard to deal with.
Yeah, that's you're pointing out a very, very important point. Even current indication, there are a lot of people who are not qualified to get the drug, and their disappointment is huge because they expect now we have something else to be better for them, and said you are not indicated for that. So very heavily mentally shocked. So how we with the physicians talk to them, this gets very, very serious and important issue for us to tackle on. In everyday clinical life, it's going to be very tough to deal with. I think it should be really stated that most patients with Alzheimer's disease wouldn't qualify for treatment. So the majority of patients, for many different reasons, wouldn't qualify for the treatment. And this patient group is, of course, a patient group which is there and which will have, as you said, a lot of disappointment and a lot of arguments and giving the doctors a tough life. It's hard to understand that there's a treatment out there, but you can't get it. Yeah, but Dan, it is true now. At least treatment is here, and so the current regulation, after experiences of a lot of cases, may be removed, or there are several ways to talk to them about the possibilities in the coming days.
So thank you for your wonderful presentation. I'm Nobu Hattori, Juntendo University. I'm a neurologist. So many neurodegenerative disorder pathologies show abnormal protein aggregation. So in this point, Alzheimer's disease, especially lecanemab antibody treatment, could be a breakthrough for neurodegenerative disorder. But so many antibody treatment clinical trials are ongoing, but only lecanemab could get successful solid. So how do you feel about this discrepancy or difference in the result? Only amyloid protein could be successful. So do you think about this point?
Yeah, thank you very much, Professor Hattori, and I should greatly be appreciated your leadership and guidance to us in a day-to-day setting. Thank you very much for that. And Professor Hattori is, of course, AD and PD specialist. Thank you very much for your guidance. Always appreciate that. And thank you for the questions. I think firstly, probably the target protein, A-beta protofibril, is still soluble oligomer and have strong neurotoxicity. This target protofibril is the right one. And also then the basic characteristics of this antibody would be okay to continuous treatment. Probably, Professor Hattori, antibody, anti-drug antibody, which is probably several antibodies got their neutralized antibody after a period of treatments. And the lecanemab case, that occurrence of neutralized agent is very low. So this is a basic characteristic of the antibody of lecanemab. So we have some very special, feature for this drug, differentiate from other antibodies.
Thank you.
Thank you so much for the great talk. Thank you so much for attending and for the great discussion. We are over time, and unfortunately we need to stop. So thank you again.
Thank you very much.